Speaking of that, guys, we've rolled up onto 11, so people will continue to trickle in. Brenda can let them in as they come in. But just for the sake of time and making sure that we stay within 11 to noon or so, we'll go ahead and get started. First off, I just want to thank everybody so much for taking the time to show up and listen about some of the important work that Dr. Sudeep Chandra is doing. We just got a little bit of a taste of that, but we'll go into a little bit more specifics coming up. Before we do that, though, I'd like to do a quick introduction to the PME team, just so you know who we are and what we do. I'll go ahead and start us off. My name is Chandler Smith, and I am part of the sales and marketing support team at PME. I have been with the company for a little over 2 years, and am mainly responsible for the creation of case studies, videos, and reaching out to past clients to make sure that they're happy with our instruments and if there's anything that we can do better for them. Ted, go ahead and give yourself a little bit of an introduction.
Hi, good morning. My name is Ted Lucena. I'm the sales account manager here at PME. I've been with the company for about, since 2017. It sounds like a long time.
Brenda, go ahead and give yourself a little.
Hi, everyone. My name is Brenda Hernandez, and I've actually been with PME for a combined 6 years. I love it here, and I'm super excited to be here, super excited for you all to join us for this special talk. And if you guys want to share any research projects, just send them down our way to sales@PME or directly to Chandler or myself. And we'd just love to hear about your guys' research.
And to wrap up our PME introductions, Peter, go ahead and give yourself a little intro.
Sure. My name is Peter Frazier. I've been with the company since early April. I'm the product technician and customer support. So I handle all the customers who come to us with MiniDOT's questions and any advice they need in deployments, or if they're having any issues with their products. I handle other engineering aspects here at PME as well. Still learning a lot, but it's going well. Thanks for coming.
Perfect. To give this meeting some context, I hate to repeat myself, but just to let everybody know, today we're going to be talking about Dr. Sudeep Chandra's work. It involves the use of the MiniDOT Clear, and correct me if I'm wrong, Sudeep, also the normal MiniDOT as well, I believe. So we're going to be learning about his research and how the MiniDOT, both the MiniDOT and MiniDOT Clear, have helped him. But I will leave the introduction of the MiniDOT Clear and introduction to Sudeep to Ted. So I'll go ahead and give.
Yeah, hi everybody. Real quick, here's a look at the MiniDOT logger. It has this white pressure case for those that are not familiar. I'm trying to do this as quickly and non-salesy as possible. 2 AA batteries, on and off switch, USB communication cable that gets you out to your laptop or your PC prior to your deployment. That USB communication cable allows you to change the intervals before your deployment. After your deployment, that's how you would download your data. The front of it has the membrane right there. Any dissolved oxygen or dissolved oxygen that lands on the membrane, the optic sensor right underneath it will measure the DO and temp. It also comes in the new clear logger, which Dr. Chandra will be talking about. Just has a clear case instead of the white case, has an LED screen that shows you the dissolved oxygen and temperature at every interval. And if you foresee biofouling to be an issue, we also have the wiper and brackets. Here's the wiper with the little brush on the side there, and that connects via USB communication cable to your laptop or PC, so you could change the intervals of the wiper. Our very special guest today is Dr. Sudeep Chandra. He's been with been a PME customer of ours since 2016, I believe. Been very busy the last few years, so thank you, Dr. Chandra, for using our devices. Dr. Chandra is a limnologist out of the University of Nevada. His lab studies lakes and river ecology. He has long-term research sites in Northern California outside of Mount Shasta, where they continue long-term monitoring of two lakes, Castle Lake and Cliff Lake. They also try to understand the influence of global chance-related issues to tropical lakes and rivers with field sites in Guatemala, the Mekong River, and Lake Tahoe. He'll be talking about their work using the MiniDOT loggers to understand changes in water quality and metabolic changes from wildfire smoke and species invasions in Castle and Crater Lake. Ladies and gentlemen, Dr. Sudeep Chandra.
Great, thanks for having me. I am still trying to share my screen here. Brenda, would you mind giving me a little boost here possibly? We can try it again. It says, there we go. Thanks so much. Perfect. Hey, well, thanks for having me. I do appreciate giving science talks and sharing a little bit of the work that we're doing. And I'm just looking forward to telling you a little bit about what we've been doing with the PME MiniDOT loggers and also kind of the science themes that we work on here at the university. And so I'm going to try to walk you through in the next 30 minutes, kind of a rapid introduction to the utility of MiniDOTs that we've been using, and tell you about kind of high frequency monitoring that we've been doing and the utility of what I like about each model. That's usually a nice way just to start off, I think. Share with you for those of you who need the punchline. And then I'll tell you some about the long-term ecological research we're working on at Castle and Cliff Lakes in Northern California, and also assessing the effects of wildfire smoke on habitat-specific processes in lakes. I'll take you up to Crater Lake up in Oregon, where we're working with the Park Service to evaluate invasions of crayfish and their effects on the lake. And I'll throw you some opportunities at the end. Hey, if those of you on video, can you raise your hand if you've used these loggers before? Or kind of curious. Okay, great. So I'll give you a little flavor for what we're doing then. So we, as Ted just mentioned, we have only been using these loggers for 4 or 5 years or so, and have found them to be really useful in all of the different research sites that we have for either monitoring water quality, just straight up temperature and oxygen or light dynamics, or monitoring that water quality and then calculating metabolic process in lakes. So that is how much production we might have. And we have a whole bunch of loggers that we are now using we work with different agencies and or working within our lab group in Northern California and Southern Oregon and lakes from Crater Lake down to Lake Tahoe. We've also been using in Nevada these loggers with Summit Lake in Northern Nevada with a tribal group there, the local indigenous Paiute tribe, to understand the water quality changes and metabolism in their rivers. Then work in Chilean rivers. They work really well in the subarctic, I think, in the cold temperature waters, and that's been helpful, along with some of the winter limnology that we've been doing in Mongolia, and then tropical work in the Mekong. So we've had a good 5-year run with the loggers being placed in different sites to contextualize some of our scientific research. I'm just giving you the punchline now of what I like about the loggers. I do this from the get-go, and then we can talk science. The traditional white loggers I really do like. They're a little cheaper by $100 or so, nothing substantially crazy actually. And so a couple $100, and so not a big deal, but they are a little cheaper. They're easy to set up and deploy. I call them student proof, especially with my undergrads. Like it's just, as long as we can do some very basic software programming and flip switches and check batteries, that tends to be the thing people don't check, is the batteries. Then otherwise you can use them fairly easily, and they're fairly for us precision sufficient, I call them, for modeling for what we want to do. And that anti-fouling wiper we've only started using recently in the last couple of years, I think the last year in particular, and that's helped us a lot. And it's just an easy design. The clear loggers I really like. They're slightly more expensive, but not out of touch right now if we're writing grant proposals or scholarships with students, and they're really easy to set up and deploy, same opportunities. The thing I love about the clear is is if we do research and we put these out in the environment in our lakes, like I'll show you, although I may not have a lot of photos of the clear loggers, but we're using them. When we have dive teams or snorkelers go out into water, then you can actually, Ted's holding it up now, you can actually see whether it's logging or not, and you can actually record temperature and stuff in your lab sheets or underwater or in your river experiments. I think there's something tangible to knowing and seeing a screen, knowing that you're actually collecting data, and then also if you understand your system, you'll know, okay, the logger is reading a reasonable reading. And so I think that having that screen with the clear loggers is great. The other part I found that's interesting, particularly in our Mekong tropical work, for whatever reason with the students there, just knowing whether or not the loggers are taking on water or not taking on water, having the clear shell is nice. It's this reassuring feeling that everything is sealed up. Not that we've had any major issues, but there are little things like that that I think go a long way for me with setting up sensor stations. It bugs me when we have other sensors from other places and we just don't know whether they're working or not. And then you come back 6 months later and you're hoping they're working. So I think it's a great design. And again, it works with the anti-fouling equipment. I really like the MiniPAR. We utilize the MiniPAR for research to look at light changes, as I'll talk about in a minute with wildfire smoke effects, but also to calculate our metabolic process in lakes and rivers. We need light dynamics. So it also is very easy and student proof. And so you can get things, they're reliable, the classic kind of PAR sensors, backbone that is very reliable. We can use them underwater and in the air. That's really, really nice, whether we face them downwards or upwards, so we can look at different light transmission rates. Now, this is to the PME group. We really need an ultraviolet light logger. If you guys can please get in there and get a reasonably priced ultraviolet light logging system, that would be great, because the current costs are out of control from some of the companies and it's just not tangible. And ultraviolet light is pretty important for tracking global change issues.
I'm writing it down right now.
Thank you. Thank you. So that's kind of where we use the probes and the things I like about them. The punchlines are out of the way. Let's get into the fun science stuff here for the next 15, 20 minutes. So what are we using these loggers for? I run this long-term research site in Northern California, but these sites that we try to study across the Sierra are really sensitive to shifting snowpack and climate forcing and climate change issues. And for those of you who are in the West, you'll know this more than ever in the last half decade to decade. We've been in a long-term drought, and therefore snowpack is also changing, which means the runoff potential within watersheds into our lakes and rivers is changing. And these lake and river sites up in the mountains, whether they're pristine or not, are really important to the economic balance and function of California, for example, and parts of Nevada, just the water resource balance itself. So we've been wanting to ask these questions throughout the Sierra Nevada mountain range. Here on the left, you can see the white, the snowpack peaks if you're not from California. This is the Sierra Nevada mountain range with my pointer pointing through it. And we've been wanting to ask questions now of, in the wake of shifting climate and or big changes from drought to pulsed water disturbances, which lakes are going to be more resilient to this change? Which systems proactively can we move into a conservation world, for example, of managing for certain species versus others, or which ones will retain their water quality? And I think this is sort of the next design of phase studies. Like, we shouldn't just be studying impacts, we should be thinking of the future and asking, do small systems versus large systems exhibit some sort of resiliency to water quality and biological change? And what's their utility for maintaining biological species? That's particularly our interest in these systems. One of the things that comes up in our talks, and this is more often than not with policymakers, is there's obviously a lot of modeling going on understanding climate change and climate influences on systems. Rightly so, there should be a lot of modeling. It's the scale dependent stuff that we're mostly interested in these days. We're trying to understand where these impacts are going to occur. But there is something tangible to actually seeing data that's not modeled. And there is something tangible to policymakers actually seeing graphs from particular systems. And I think for making policy level changes, having sensors placed into networks in these mountain systems become more and more important. It turns out in California, in these mountain range areas highlighted in blue, there's only 2 sets of lakes that have long-term monitoring associated with them. 2 lakes in the, or a set of lakes in the south, the Sierra Nevada Tocopah Basin, have been monitored from 1980 to present by my colleagues Steve Sadro, John Milak, and others. And they've just done an excellent job of anchoring these lakes in the southern basin. But our group has been measuring environmental change in these mountain lakes in Northern California at Castle and Cliff Lakes historically since 1959 to present by Charles Goldman's group and now ours. And so believe it or not, in the state of California, for all the talk about how important climate change is going to be, there's only like 4 or 5 lakes that are these anchor lakes for understanding climate change empirically derived from these systems. I'm going to take you to 2 of these lakes, Castle and Cliff Lakes, and some of the small lakes surrounding them. Absolutely beautiful. It's the garden spot of the Siskiyous. You really should come up for a visit if you would like to and see these sites. Just absolutely wonderful, gorgeous systems. Castle Lake is a 35-meter deep lake, and it has a nearshore habitat that's pretty dominating in the lake. About 54% of the lake is nearshore or littoral zone is what we would call it in limnology. And it has about a 2 to 4 year residence time. So it's very sensitive to changes in snowpack and watershed changes. And its nearshore and offshore habitats also become very sensitive to changes. And so that's kind of what we've been interested in. So what do we do at Castle Lake? Well, we work on a number of things since 1959. We have measurements that we use from different agencies and the Forest Service, so on and so forth, with snow, water, ice content, snow depth, air temperature, wind, and solar radiation. And then since 1959, about 15 to 18 times in the year, mostly in the summertime, sometimes up to 20, we would measure temperature, oxygen, the physics of a lake. Then we measure the chemistry of a lake, the productivity of a lake, and the biodiversity. And what we've done is we moved from using, we use not only profiling units from other companies, but we've actually started to put in fixed station MiniDOTs at different depths in the lake. So we can track oxygen and temperature changes in the different strata of the lake to try to relate it to the snowpack and snowmelt or the drought conditions. We also use these MiniDOTs at fixed stations, as I'll show you in a second, in the different depths of the lake or in the nearshore or the offshore to try to calculate metabolic productivity of the lake. And this becomes increasingly important as we have changes in drought. We've changed the food supply going into the food web for fisheries, for example. So what is this deal with climate? Why are we so worried? Well, these mountain lakes in the Western United States and globally traditionally have winter ice pack on them. And the ice pack is a way that maybe the system, I guess in a way, can reset itself, but it's also a dynamic control of the light going into the system. So Castle Lake actually has one of the longest running ice out data sets for a mountain lake in North and South America. There's so few empirical data sets. In fact, we actually couldn't find any that go back to 1959, where the study I'll just show you. So it's kind of sad, but you'll notice over time that the ice out is highly variable by Julian date. It could be as late as July, early as March, and as late as, so early as March or as late as sort of July-ish. And what you'll notice recently is these ice out dates are getting pretty wacky. You're getting really early ice out or you're coming back to normal. Not unexpected with drought conditions, but things are changing in my recorded lifetime for sure up there. And so what we wanted to try to understand now is what is the influence of these different ice dynamics set up for the growing season, for the algae growth that's in the different habitats, the oxygen and water quality data, or the full mixing regimes that we might see in the lake. And so we started taking advantage of these early, mid, and late ice out dates and starting to ask questions of like, what happens to the lake's water quality? What happens to the full mixing of the lake? And how does this affect trout feeding behavior? I won't go into the details of that work to date, but some of it is summarized in a recent paper by my graduate student, Tim Caldwell, in Global Change Biology, where we've tried to look at climate and snow impacts into ice phenology and breakup. And then through the water quality measured through the MiniDOTs and vertical profiling, how might it affect production in the lake? And then also how might stream flow entering lakes affect how fisheries behave. And so you can see that paper in Global Change Biology if you're interested, or I can send you a copy. So coming back to ice dynamics and climate, remember I just told you that these mountain systems are becoming increasingly more sensitive to climate. Yet they're also a bunch of small lakes. They're not large lakes and they're really high in elevation. So there is some resiliency to these ice out dynamics. And in fact, if you look at trajectories of ice out to date for lakes across the Western United States in the mountains, not in the Midwest, but up in the mountains, you really don't see trajectories changing just yet. You're seeing instability in the ice out date, like I just showed you on that graphic for Castle Lake. So what my student did, this is Tim's work as well, really grateful he kind of pulled this off. But what we ended up doing is using remote sensing and meteorology data to then look at small lake ice out dates for a series of lakes from the Cascades down to the Sierra Nevada. This little triangle is Castle Lake, that little lake I just told you about. And it's our anchor lake for these studies for some of the data. And what we ended up finding, I'll give you the punchline, and this paper was published in Limnology and Oceanography last year. What we end up finding is that these ice out dates are generally driven from the climate models, are generally highly variable across all the lakes. They're slightly declining or occurring earlier. But if you use kind of the business as usual climate models and force them into these climate projection ice out models that we made, you're basically going to see ice out occurring 50 to 60 days earlier. And I'm a pretty big optimistic person, but my feeling is in my lifetime, we will be business as usual. So here we go in this sort of ice out dynamic is going to occur 60 days. So why do I care about the lake then, right? If it's going to occur 60 days earlier, what's going to change? Well, 60 days earlier means that there's sunlight occurring 60 days earlier. It means that the balances of the water quality might shift in terms of how the lake mixes and the temperature and the oxygen that you might see. So having fixed sensors identify and tell us what these relationships are going to be really important. So here's some work that we've done with the MiniDOTs, for example. Our students are using the MiniDOT clears, and we've been putting them either under ice in some lakes, like at Castle Lake here shown in the bottom photo, or this is our student Josh Culpepper, who's doing some great work of some small lakes in that same neighboring region, and we'll anchor these clear water MiniDOTs in the lake. And so now we can let them run from summer to winter back to summer and see what ice dynamics do. And I'll show you just a little bit of the data. Apologies for how complicated this might look, but in short, you've got not Castle Lake. Oops, I put 3 of the same lakes on here, but you've got lakes that are smaller than Castle, not 35 meters. And what you can see is the oxygen change in this left part of the graph in March, April, May. Some of the lakes are exhibiting drastic drops in oxygen in what should be winter, but then they rebound in wintertime under ice. To us, this was a big surprise. Under ice, oxygen is now, because of the MiniDOT detections, are basically coming back up even when they're under ice. And so there must be these watershed subsurface flows that are occurring. That's what we're guessing, an aerated water coming in that drives these oxygenated environments. Versus if you go just to the neighboring lake, one watershed over, one small watershed over, the oxygen in the black here remains depleted over time. And there's a slight jump in the oxygen. It's still pretty low, but there's other physical controls going on. So ice is not just a cap on lakes, but the connectivity of whatever's happening to internal process in these lakes in wintertime are very important. Our colleague that I'm learning a lot from about temperature and oxygen dynamics, Dr. Sally McIntyre at Santa Barbara, tells me there's a lot of internal lake processing. And what I love about that internal lake processing, it means that the under ice process becomes more important for controlling oxygen and temperature dynamics. So why do we care? You're measuring oxygen and lake dynamics. Well, if this is at Castle Lake, that big lake I told you about, it turns out oxygen, as those of you who are chemistry oriented and physics oriented, oxygen at the bottom of lakes is a large controller of the type of nutrients that are available. And the type of nutrients, of course, would drive the types of summertime productivity that you might have later when the lake mixes. And what our student has done is Josh has gone out and he's used these MiniDOTs, placed them at the bottom of Castle Lake, for example, at 20 and 30 meters depth. And now we can tell that across seasons, in years that are a little more wet versus dry, so what you can end up seeing is that in years that have earlier ice breakup, so let's say the 60-day window earlier or 30-day window, the oxygen actually declines a lot faster in the bottom of the lake at 20 and 30 meters compared to the rate of change when the ice breakup is more average. And so these oxygen controls just for a month period become pretty important for thinking about for the month to summer periods become very important for controlling nutrients that would be available, or maybe where fishes live. And so these fixed station probes are allowing us to then get at depth specific changes at time scales that are relevant for ecologists to actually measure. I'm going to move on briefly. Any questions so far?
I think that might be something we want to save for the end, just so everybody can kind of collect their thoughts and then, but great point to bring that up. Please, everyone, feel free to use the comment section function at the bottom of the screen, the chat, I guess you could call it. Feel free to use that to collect your thoughts there or post any questions that we can address at the end.
Perfect. So we're going to then move on into thinking about, it's one thing to think about ice dynamic changes and their effects in the bottom lake habitat, oxygen and temperature changes. But now we've been wanting to ask, well, we have a lot of fires these days, and these fires are putting up a ton of smoke. And how are these smoke changes affecting lakes in their short and long term? And in this particular case, we're asking not the water quality itself, but how might it affect the productivity of a lake that we can determine from the oxygen sensors. And so this is Castle Lake again in the center. And these are the 2018 fires, for example, that were occurring within a 150-mile radius. And you can see from the upper left graph what Castle Lake looked like under some of these changes. There's days in non-smoky years when it's absolutely beautiful, and then there's days in smoky years where our basin is getting blanketed with smoke. And so light is being reduced. Ultraviolet light too, as you'll hear about in a second, that we want the PME guys to make a sensor on. And so we put out oxygen probes in the nearshore, we call them the littoral zones, and then we put them out in the offshore because you're going to have bottom algae versus open water algae and bacteria producing food in the system. And this is the work of my colleague and postdoc, Dr. Facundo Scordo, and this is sort of hot off the press, actually. We're about to submit this. And what we're basically finding is in that lake, when you have these extensive wildfires, of course, your air quality goes down. And this is particulate matter 2.5 air quality that we have measured in the basin in 2018, compared to 2015 to 16 and 19 in years without smoke. You can see that the air quality is much better. So not a lot of smoke in the basin in those controlled blue years. But what you end up seeing is that light control has a dramatic effect on the lake. It changes the heat content of the lake. So we use the temperature data from the MiniDOTs to then and other forcing variables to calculate heat content. And what you end up seeing is that in smoky years, your heat content indeed does change in the beginning of the season compared to the non-smoky years. So the gray here is the smoke period in the lake. But what we also see is while the lake may be cooling a little bit, which means that algae should actually be decreasing, what we also end up seeing is that ultraviolet light changes dramatically during the smoke period. So the gray here indicates the smoke period days, and you can see that ultraviolet light gets highly depressed in the lake compared to where it would be normally around 0.6 meters. And then photosynthetically active radiation, or the stuff we can get from the MiniPAR, for example, is also depressed at 3 meters dramatically. And so you're really activating and changing light, temperature, and then from these smoke effects. So then what do you see with production? This is kind of a graph that calculates using from the oxygen data that's from the MiniDOTs. It calculates how much gross primary production you might see, how much respiration, and how much net ecosystem production by putting it through models. And what we end up seeing is in one habitat, the open water habitat, not the nearshore habitat, gross primary production calculated from the oxygen data and temperature goes up in the open water habitat. So smoke actually doesn't suppress production in the shallow water, it increases the algae production. And in fact, in a paper that Facundo published in Nature Scientific Reports earlier this year, the production went up 109% in the open water pelagic habitat. You'll notice that in the two nearshore sites, the nearshore edges of the lake where there's usually thick paraphyton growing, it doesn't really do much to it. The light depression in this case actually doesn't affect the amount of oxygen in the system and in the production that's being produced. So the nearshore habitats seem to be resilient to wildfire smoke changes. They maybe are more in the background reference condition compared to the open water habitat. And indeed, it also changes other aspects of the system, including net ecosystem production. So once you compensate for understanding respiration. Long story short is these probes have helped us to try to, and profiling has helped us to try to understand smoke effects on lakes and then what their relative controls might be on different habitats within lakes. And for those of you who probably if you ever want to take my limnology class, I always try to remind people that we always try to go measure this stuff in the open water. That's what managers are set to do. We usually are really used to monitoring things traditionally in the open water, but all the action is happening on the edges. So for those of us who fish, we know we like fishing on the edges of lakes because that's where the bugs are, that's where the fish are. And so I think there's this reconciliation in limnology and aquatic ecology where we're trying to understand two habitats these days, not just the open water. So please do use these types of probes to monitor multiple habitats and not just one if you can. So that's the effect of wildfire smoke on metabolic processes through oxygen. I'm going to take you a little further up north, just briefly, for the next 5 minutes, up to Crater Lake in Oregon. Quick hand raise for the people on video who've been up to Crater Lake in Oregon. Who's been up there? Yeah, spectacular location. It's our national park, dedicated to being around this big, beautiful, clear lake. It's just spectacular water clarity. And so this is an outline map of Crater Lake. There's an island called Wizard Island in the center. How cool is that? And then there's these, underwater volcanic geology. And then you have this area called Cleetwood Cove, which is where you would walk down from the rim down to the water. And those will become important in a second. Crater Lake is this just spectacularly beautiful place. We're actually heading up there tomorrow to do some more of this work. It's a historically fishless lake, but it has two fishes that are introduced. Its native predator was this Mazama newt. It's an endemic newt only to Crater Lake. It really is cute. The photo doesn't do it justice here. But it has a series of crayfish introductions that have occurred. The signal cold water crayfish was introduced in that lake in the 1915-ish period, 1920s. And crawfish were planted to try to bring and make bigger food for the fish. And it turns out our colleagues at Crater Lake National Park, while they knew crayfish were in the lake, they really didn't observe high numbers of crayfish until about 10 or 15 years ago. The numbers, for some reason, of the crayfish started exploding around the lake and expanding all around the lake. And this graph on the right side, you can see the densities of crayfish from our 2015 surveys, mostly on this North Shore or where Cleetwood Cove is, definitely around that island called Wizard Island. Again, great name, right? Not a lot in the South Shore, for example, of the lake. So we've done some studies looking at crayfish invasion up there and expansion, and we know the crayfish utilize cobble boulder habitat. These are different than the crayfish that we might see in the Midwest or the South, for example. And so boulder habitat becomes very important for this little crayfish that's the size of your hand that lives to about 10 years old. What happens where you see areas with and without crayfish? So the northern sites versus maybe the southern sites. Our student John Umek went out and measured invertebrate biomass, so snails, chironomids, worms, and basically found in areas where there were crayfish absent, we always saw higher densities of invertebrates. So the things that would graze algae or eat each other, right? But that where there were crayfish present, we saw a big depression of these snails, of chironomids, of worms, of the things that would usually eat and graze down algae and keep the rocks all clean. So what's the influence then of crayfish on the algal actual production and changes to the invertebrate densities I just showed you? Well, if you do a little experiment, this is not field data because we're working in a national park. This is kind of, at first we started off with a very basic science project. In areas where you have very high crayfish density, you have very little invertebrates, and you have lots of algae growing, green patches. And then in areas where you have very little crayfish consumers, those crayfish are not eating all those benthic invertebrates or gastropods, snails and things, and you end up having less algae here. And so these were first experiments done about 5 or 6 years ago, getting us to think, not from a monitoring viewpoint, but from a very simple park management viewpoint. Are we going to grow more algae with crayfish or less algae? And indeed, we thought we were going to grow more algae at a faster rate. So what we did is we've been working with the Park Service, and the Park Service has bought MiniDOT loggers from PME. And we basically put out sensors in areas with and without crayfish. And we developed a buoy system. And some of these are the white traditional loggers, and now some are, I think, are the clear loggers that we've set up. So we put a buoy in the water, we put up turbidity sensors and algal sensors, chlorophyll sensors, and then we put in the oxygen sensors in there also. And we're basically able to now look at, I'll show you a photograph, it's much better. Subsurface float, where we can now monitor oxygen and temperature and water clarity all year round underwater. And this has been quite exciting for us because the park can only monitor the lake from June to September safely based on weather conditions and wave conditions and access. And so now we're getting data all year round underwater during winter. And here are what some of those graphs look like. So again, this is a model of the oxygen data. And the sites aren't going to make too much sense to you, but I just wanted to show you the type of data that we might get out of these systems from 18 to 19 in different locations. And so this is gross primary production. Again, the amount of food available at the base of the system, let's say, in a lake. And indeed, what you end up getting at sites at Cleetwood, which is in green, which is for those of you who hike down to Crater Lake, it's the area that you could hike down to the waters where there's the largest crayfish invasion. You end up seeing very high gross primary production. And then at sites away from crayfish invasions, the yellow, you get less oxygen change and less primary productivity. And so this is indeed the summary of what we've been finding. Again, this is also not published. This is just sort of hot off the press, and we're looking at submitting this here in the coming 3 months. Pretty excited about this data, but basically Cleetwood, in this case, I don't even know what you call that color, peachy color, you get clearly higher summer gross primary productivity where there's crayfish, and then where there's no crayfish at Eagle, you get much depressed productivity. You also get changes in the type of respiration that you might see, whether it's from photosynthesis and or bacterial production. And so these crayfish are having significant impacts into our national park where they've invaded, and they're changing the structure of the greenness of the bottom of the lake and the amount of production, not just the standing biomass that you might see. So that's kind of a whirlwind tour of our sites in Northern California and Oregon. I would have loved to talk about our work in the Mekong, but could save that for another time. What I wanted to highlight and do a plug on is there are a couple of papers that we've been involved with recently that just beg for the use of trying to understand high frequency information and sensors to try to understand changes that are occurring to our pristine lakes or changes that are occurring to our lakes globally. And so here's some papers to check out. The first one was just published in Bioscience earlier this year. And we're finding that pristine lakes globally are starting to have these nearshore benthic algal blooms, whether it's like at Crater Lake or up at Lake Tahoe, or maybe because of this wildfire smoke, it's not entirely clear, but this is happening globally. And I think having these nice, simple handheld sensors are going to be really important for documenting the mechanisms of change in the future in these pristine systems. Then there's another nice paper led by our colleague Kevin Rose and his student Stephen Jane in Nature that was just published. And we just published this in June, and it's showing the rapid deoxygenation of temperate lakes. There's actually some tropical lakes in there as well in that data set. And this is, I think what's happening is these papers are describing big changes, but sensors and other studies need to be put out into these systems so we can actually understand the mechanisms behind some of these changes that are occurring. And I think that's it. That's all I have for you.
Wow. Thank you so much, Dr. Sudeep Chandra. I really appreciate it. So as mentioned before, there is, doesn't look anybody posted any questions. There was also a lot of information to sift through. So I absolutely understand if people want to think through all of this and maybe formulate some of their own questions. If that is the case, is there some way that they can reach you, an email or something of that nature?
Totally. I'll put my email in the chat, but you can also just look me up at the University of Nevada and just type in limnology, University of Nevada. But you're more than welcome to, especially the early career folks out there, you're more than welcome to just, schedule a time, come take a trip up to Northern California and see some of our sites, or happy to correspond with you. I just can't tell you how delighted I am when we use these types of high frequency measurements to try to understand change and with mechanistic changes that aren't just these grab samples once a month and or instruments that are difficult to use. So I'd be happy to guide you through what we're doing.
Yeah, I have to admit, I am pretty jealous. You're doing some very, very important work in some of the most beautiful places I've ever seen in California and up in Oregon. So like I said, I gotta admit, I'm pretty jealous. But before I let everybody go, is there any last minute questions that anybody has for Sudeep, for us here at PME? If not, I'll go ahead and let everybody go and thank everybody so much for taking the time to listen to Dr. Sudeep Chandra and the important work that he's doing. And enjoy the lunch on us, guys. We really appreciate it.